knows that insects can navigate and return to their home nest. But how extensive is that memory? How long does it last? And which part of the brain controls it? The fruit fly, Drosophila melanogaster, was used by Tyler Ofstad, Zuker and Michael Reiser in the paper Visual Place Learning in Drosophila melanogaster to determine if flies were able to form reliable spatial memories in complex environments. This was investigated through the use of coloured tiles of which the temperature could be changed and light-emitting diodes or LED lights. These were set up in a circular chamber in which the flies could be contained. All the tiles were heated to 36 degrees, except for one, which was heated to 26 degrees. As the flies favoured the cooler temperature, they congregated on the cooler tile. The LED lights were manipulated to form various constant visual patterns that could be moved to different positions on the tiles. The flies were then trained to recognise the cooler tile that was kept in a constant position in relation to the spatial and visual surroundings. Once the flies had established where the cool tile was, all the tiles were heated to 36 degrees, that is, the hotter temperature. The LED light patterns were also rotated by 90 degrees. Despite the loss of the cooler tile and change in visual, the flies explored the previous position of the cold tile for about a minute before giving up. That the flies returned to the previous location of the cold tile indicates that they have the ability to form memories in relation to spatial temperatures and visual surroundings. After it was established that the flies could form these memories, they were taken from the testing area. They were then returned to the testing area at different time points to determine how long the fly's memory would last. The flies would return to the old position of the cool tile for up to 2 hours after it had been removed. This demonstrated that the flies could remember things for at least 2 hours after they had been removed from stimulus. The ellipsoid body is the part of the brain that controls movement and other tasks in flies. After a few tests, it was evident that the use of fly lines containing silenced neurons in the ellipsoid body made flies less able to use visual place learning. This allowed the investigators to determine that it was the ellipsoid body that controlled, or was at least related to, spatial memory. The article, Visual Working Memory, Now You See It, Now You Don't, by Lily Karsai and Troy Zars provides more extensive studies into the visual working memory of fruit flies. It also details the ellipsoid body as being responsible for visual memory and investigates how the gene ellipsoid body open influences the normal working memory. I hope you have gained an insight into how memories of Drosophila melanogaster work. Thank you.